Hi everybody, welcome to the next video on physiology regarding salt and water balance and how we do that and how we excrete the waste products that our body makes. I put this picture in here. You're probably wondering, why is there a woman on a camel for this introductory slide? I noticed that the camel is actually urinating. So I thought it was a very appropriate picture to show, even in desert conditions where there is very little water, the landscape is very, very dry, animals still need to exchange water. So that I thought the camel was doing a great job illustrating that, right? And so if you recall that animals, again, we must use water in order to make exchanges of the materials within our body, the nutrients, the waste products. And so recall that single-celled organisms used to exchange directly with their environment, but they lived in a watery medium. Then when we get multicellular organisms, where we have more and more cells that are further away from the external environment, that exchange just through diffusion is impossible. And so we created body cavities. Remember that? And we put water inside of those body cavities. So we're kind of creating our own environment within our bodies. That's our internal environment. So therefore, with animals, water is the major portion of the animal's weight, of the body mass of it himself. All of our cells, remember, have water inside of them, just like the single-celled organisms. But then, different from the single-celled organisms, remember, when our cells work together and form tissues, then there's fluid surrounding those tissues, right? That was the interstitial fluid. And then even more complex animals created a circulatory system to help move the body water around and exchange with the interstitial fluid and with the cytoplasm. So that's the transport medium, because we need water in order to transport dissolved nutrients and minerals and other things that we need, as well as waste, right? And so recall then, one of the other things is electrolytes. So we do have salts within our body, and they dissociate into charged ions. So when you consume sodium chloride, which is table salt, when it gets mixed with the water, it will dissociate into an ion of sodium, positively charged, and an ion of chloride, which is negatively charged, right? And so we have to keep that balanced, because since those separate and they can move to different sides of a cell membrane, that imbalance can alter the membrane potential. Well, we already learned in the nervous system that we do that on purpose. We manipulate the balance and charge across the membrane in order to change the electricity, right? And have electricity move down the membrane, and we use that as a signaling molecule. Right, but other cells, we don't want to change the polarity of our cells. So we have to maintain a balance of salts, and we do that through the regulation of the salts as well as the water, right? So dehydration, and this picture kind of shows, dehydration is a very serious problem with animals. Dehydration really compromises the circulatory system, therefore the movement of nutrients and electrolytes and everything we've been talking about, and waste products, and it also upsets the regulation of body temperature as well. So remember that animals, we always need to be able to balance that water, and we may have to replenish the water consistently. Okay. Here's another slide that just illustrates a little bit more what I had been talking about. So again, remember that intracellular fluid, or the fluid inside of our cells, the cytoplasm, that holds most of our body fluid, because our bodies are we are multicellular. We're made out of cells, right? And then, so this picture to the right of the slide is showing two cells here just to kind of indicate a multicellular organism, and that would indicate a tissue. And so on the outside of those cells is the body fluid called interstitial fluid that just surrounds all of our tissues and things like that. And that might be in coelomate animals. That's what's contained in the body compartments. And then way over to the right, it shows a blood vessel. So that's another compartment holding water, right? And the fluid portion of blood is the plasma. Okay, so you can see with these arrows is that the water from the plasma can move into the interstitial fluid and can then move into the cell, right? But it can also move back out into the interstitial fluid and back out into the plasma. So water is constantly being cycled around these three compartments, okay? But so remember that the fluid is also carrying salts and other solutes. And so changes in those salt concentrations can lead to changes in the fluid distribution, right? So if the cells are surrounded by fluid, the interstitial fluid, that is saltier than it is on the inside of the cell, water is going to move to the area of salt. So that means water is going to move out. And that's what you see over to the left side of the slide with those three red blood cells. Okay, so the red blood cell we're using as an example of a typical cell in our body, right? And so just imagine in all the white area, that would be interstitial fluid. Okay, well, the salt concentration or other solutes like sugars and things like that, if that is equal to the concentration of the solutes inside of the cell, then we say the two are isotonic to one another. So that red blood cell at the top is sitting in an isotonic solution because the red blood cells are, or the salt solution is equal inside of the cell as well as outside of the cell. So water is still going to pass through the cell membrane, but it's going to do it at an equal rate. So therefore, we have an equal concentration of salt. We maintain the balance, and we maintain the balance of water between inside and outside of the body. But let's say if you follow the arrow down to the left to where it says hypotonic solution. Let's say you were given an IV of pure water. What we're doing then is we're putting an intravenous solution of pure water into your plasma, so your plasma has less solute in it. We've diluted the salts and other products in there. So your solution, the plasma surrounding those red blood cells, we say is hypotonic. Hypo means less, and it means less solute. It has more water in it. So it's less solute than what? It's less solute than the solute concentration on the inside of the cell, right? So remember, solutes are things like salts and sugars and that kind of stuff. Right now we're just talking about salt. Okay, so in this situation, there's more salt inside of the cell than there is outside, which means there's more water outside. So water's going to be attracted toward the salty solution. So water, because our animal cells are permeable to water, water is going to move into the cell. Our animal cells are not permeable to sodium or chloride unless we have a transporter there, right? And you saw that in the nervous system. If we had a transporter, then we could move sodium across. But so in any given situation, when there's no transporter, as is in the case here, only water's going to move in. And so since our animal cells don't have a cell wall, our red blood cells can actually puff up, enlarge, and they could actually burst, right? So we're using the example of a red blood cell, but this is true of any of our cells in our body. Okay, now let's say you were given an IV solution of, say, 10% salt. That's a lot of salt for our body, considering our normal salt is about 0.9%, right? So now if your blood plasma, so the fluid surrounding your red blood cells, your blood plasma is is hypertonic, meaning it has more salt solution than inside of your cell, then that means the water, again, is attracted toward the salty side. So water is going to move out, and you see the arrow of it moving out. And again, with our animal cells, we don't have a cell wall. So without the water kind of providing structure to the cell membrane would collapse on itself. Our cells would crenate or shrivel up, right? And then they're useless. They can't carry oxygen like they do, and they would be harmful to our body. So our bodies are constantly exchanging water and exchanging solutes in order to make sure to maintain the appropriate solutions within our different compartments, within the cell, within the interstitial fluid, and within our blood vessels. Okay, and this is true. So mostly here I'm talking about closed circulatory systems, but it's the same with the open circulatory system. So I'm going to show you some examples of invertebrates as well as vertebrates. Okay, so what we're really talking about is obligatory water exchange. So we have processes in our water, in our bodies, that require us to either gain a nutrient or solute or excrete it or to get rid of it. We have to use water in order to do that. So that's called obligatory water exchange. If you're obliged to do something, you have to do it. If you're obligated to do something, you have to do it. So that's where the word obligatory comes from, right? So again, these are vital processes in our bodies, but they disturb water exchange. They disturb the water balance, right? So we're going to talk today a lot about nitrogenous waste excretion. So with the nitrogenous waste, our cells, when they're doing the different work that they're doing, especially maybe making DNA, exchanges with DNA, making proteins, right? We end up making a waste product with nitrogen in it. And we can't tolerate that in our body for too long. It gets toxic. So we have to excrete it. And again, a certain amount of water is going to be required for that. Okay, so we're going to look at how that occurs. Even exchanges during respiration, in order to get oxygen into our cells and release the CO2 out of our body, we lose water doing that. So you have water vapor in the air, also the water associated with our mucous membranes down our trachea and our respiratory system. As we exhale and the air travels by it, we lose water in the process. Okay, we tend to gain some water when we eat food, okay, and also salt. So that added water and salt can actually upset the balance within our cells. So we need to deal with that. Okay, regulation of body temperature. When you sweat, we lose water in the process, right? That's what sweating is. Water holds heat, it traps heat. So when we sweat, we put water on the surface of our skin and then it evaporates away. It pulls the heat with us, so that cools us down, but we lose water in the process of doing that. Okay, an animal that pants, right? Your dog sticks his tongue out. What it's doing is creating air to go over the moist membranes of its tongue and its mouth, releasing air, releasing water, which is taking the heat with it. So that's evaporation occurring there. And you'll see different animals do that. Sometimes you see birds opening their mouths and they do what's called gular fluttering. They'll see their neck go like this. And what they're doing is they're rapidly passing air past those moist membranes in their throat so that the air evaporates out their mouth, taking water with it, taking the heat with it. So we lose water. Okay, so we have to balance water. And you saw in the previous pictures here, let me go back. Oops, that's the one I wanted. These guys are drinking water, right? So we're constantly replenishing our body with the water loss. But again, as that water, we're either losing it or gaining it within our cells and our tissue cells here. We have to balance it because of this exchange between the different body compartments, right? Even that camel has to drink water at some point. Okay, so let's look at how that happens. This is kind of a big slide, so we'll spend a little time on it. Okay, very simple organisms, like I put the picture there, the sponges and the cnidarians, they live in a very stable environment within the ocean, right? And so the salt concentration doesn't vary much within the ocean. And remember, they're more acting like single-celled organisms. So their cells, every one of them has contact with the external environment. Okay? And so they just equilibrate the concentration of water and electrolytes within their cells with the concentration in the seawater. So there's really no specialized structures in order to create that balance. And so there's very little energy expended. It's mainly through simple diffusion that these things cross the cell membranes. Okay, but those organisms that have become much more complex, again, they have thicker tissues, then a variety of homeostatic mechanisms have evolved that regulate the volume and that ionic composition or electrolyte composition of the fluids in the internal environment of the body of the organism, right? Especially a terrestrial animal, they have to conserve water. We live outside of water, so we have to have ways to conserve the water as well as replenish the water, right? So if we don't, we have to carefully regulate any water loss, and we have to replace that water loss. So we're going to see organs that do that. So there are actually two processes that help maintain fluid and electrolyte homeostasis in animals. And that's the two you see there, Osmo Conformers and Osmoregulators. Okay, so Osmo Conformers, these are most of your marine invertebrates, and there are a few vertebrates that do this. So they match their osmolarity to the let me say that again, you can read it there. They match the osmolarity fluids to the seawater. Remember, osmolarity is a term we use when we're talking about the balance of solute within water, right? That's osmoregulation, the balance of salt and water in our body. Okay, so if you're an osmoconformer, conforming means you're conforming to the external environment. Okay, so they have a less of a tendency to gain or lose water across their skin or their gills. Okay, and so therefore, again, it's just being matched. Okay, so they don't really have to expend a lot of energy in order to gain more water or excrete salt or vice versa. Okay, so that's an osmoconformer. Okay, they're just matching to their external environment. An osmoregulator, though, is actually maintaining an internal osmolarity. Okay, they're maintaining a constant internal osmolarity. So no matter what's going on in the external environment, they're going to maintain a certain water and salt balance within their bodies. That's like what we do. We have to. We can't conform to the external environment because we live on land. It's dry, right? But even those in the water do this, and usually in unstable habitats. So remember, the seawater is much more stable. So we tend to see a lot of osmoconformers there. But in estuaries, where the salt concentration of the water can be very strong at one moment with the tide coming in, and then be very dilute when the tide goes out, but rivers, fresh water is feeding that estuary, or in the fresh water itself, and of course in the terrestrial environment. So osmoregulators will have organs that can homeostatically control that optimal salt concentration and water concentration, right? But it requires a lot of energy to do that. But there's a cost-benefit analysis that we can go through when we look at these things, right? And then, of course, we need to drink water as needed, and even excrete water is needed, right? So this is that introduction into the urinary system where we excrete water as we need to, right? You'll notice yourself, sometimes you urinate a lot and sometimes you urinate a little bit. That's your body's system regulating the amount of water in there. Okay, so let's look at some of the organs that might help to regulate water and fluid. And so first, let me show you just kind of a generalized organ, what all of these organs tend to do, right? So remember, there's body fluids, and so either in the form of like us, so here's a closed system with blood. This could be an open system with hemolymph. But basically, there has to be a way to filter the fluid out of the circulatory system, right? So that's the first step, is filtration. So you can see at the top there, it's usually associated, the organ is associated with a capillary bed, okay? And that capillary has fluid pumping through it, the blood, and it's under a particular pressure, and that pressure is usually enough then to push the fluid out of the capillary into the organ. Okay, and so the fluid then that's moving into the organ is being filtered. So that's removing some of the water and some of the small solutes out of the blood. So that produces a filtrate. It's automatic. It's just because the capillary is leaky. Okay, and then this other structure is going to be designed to capture that fluid. So that's called the filtrate. And it's usually indiscriminate in its filtration. Okay? It doesn't mean it controls what goes into the organ. All right, it's just anything within that blood is going to move through, except for the cells, except for blood cells. Those stay within the blood. Okay, and you can see that not everything. It's not that the blood is draining in there. It's just filtering some of the fluids through. So you can see the fluids continue as the capillary continues down and heads toward the vein. Okay. Now, I mentioned that when it reabsorbs, or when it filters indiscriminately. So there are some things we want to keep. So we actually, most of the water that gets filtered out, we're going to reabsorb that. So that will be step 2 in these systems. Reabsorption. So you can read there the desired solutes. So those are things like our salts, right? Those are things like many of our sugars and things like that. We're going to go through with the vertebrate and see what are the things that we keep. That will come out of this organ and go back into the capillary, right? That same capillary bed, you can see that it just wraps around to a different part of the organ where reabsorption will take place. And sometimes it's active transport. Sometimes we actively transport, and so we expend a little energy to recapture the stuff that we don't want to excrete. Okay, anything that stays in this organ, it's kind of a tubular organ, anything that stays in the tube will eventually be excreted. So we don't want to lose the good stuff. Okay. However, again, I mentioned that we don't really control the filtration, so some things get by at the filtration site that we need to get rid of, right? And so that's the process of secretion. So that's a different part of the capillary where we can, it's usually variable, where we can kind of measure in our body, well, we still have maybe too much potassium, and so we will secrete it back into that tubule, and anything that's in the tubule will eventually be excreted out of the body. Alrighty, so we're going to look at those different things as we get into the different types of more complex systems. Okay, so let me go through some of the vertebrate systems, or I'm sorry, the invertebrate systems. Then I'll end this video, and then we'll go to part 2 and we'll look at the vertebrate system. Okay. So here's a very early excretory system that we find in very simple organisms. Here's our flatworm again. Remember the platyhelminthes. Very simple organism. Remember, it's acoelomate. It's so small that it doesn't even need a circulatory system. It doesn't need body cavities. Okay, but it does have a rudimentary system. We already saw a digestive system, and it also has a osmoregulation system, right? So its system is referred to as the protonephritia. So one of the structures in our bodies you're going to see in the vertebrates is a nephron. So proto means before. So this is before the nephron is how they named it. Very simple. So you can see in this flatworm, he's got a series of branching tubules throughout its body. It looks kind of similar to that gastrovascular cavity. They're not drawing the gastrovascular cavity here. So this kind of parallels it. Okay. It's just a branching system of tubules that has the ability to filter fluid from the body cavity, because at the end of each of these branches of tubules is a cell. It's a single cell called a flame cell. And you can see over to the right there, just above number 1, it's a cell. It's got a nucleus, but then the shape kind of opens up, and in the middle of the cell, there's a series of cilia. And those cilia are constantly beating back and forth. And the reason why this cell is called a flame cell is because under the microscope, that beating back and forth of the cilia makes this look like a candle. Makes it look like a flame flickering on a candle. Really, the reason why it's flickering back and forth is it's creating a current. So it's drawing the interstitial fluid through the cell, right, into those slit-like openings, and into the flame cell, which then is attached to the tubule, right? So that's filtration right there. It's causing filtration. As it travels through the tubule, remember the tubules are made out of cells, so those things that are good for the planarian, like water, some of the salts and things, it gets reabsorbed right back into the tissues of the body, okay? But what's ever remaining, the fluid that's remaining, including water and waste, they actually continue through that series of tubules, which will then open to the ends of the body in a little pore called a Nephritio Pore. It's a little hole inside of the body. So mainly, like I mentioned, those nitrogenous wastes, that can diffuse through those Nephritio pores right out of the body. Okay, so that's the protonephridium. Well, then as we get into a little bit more complex organisms like this annelid, right? This annelid, or an earthworm, which is a type of annelid, it has a tubular network system referred to as a metanephridial system. Metanephridial system. Okay, so it begins with a little funnel-like structure called a nephrostome. You see that down right in the middle between 1 and 3 there. A nephrostome. So that's a funnel-like structure that's open to the body cavity. Remember, these guys are coelomate. So, and this can happen similar in pseudocoelomate organisms. It opens into the body cavity where the fluid from the body is drawn into the nephrosome, which then gets drawn through that series of tubules. You'll see the tubules, I think of like a twisted up straw. So fluid now is drawn into the straw and travels around the length of the tubule as it twists and turns. Okay. Notice though that it also has a blood system associated with it. So the blood vessels, remember this is a closed system for these guys, the blood vessels branch and form a bed of capillaries surrounding that tubular network. So as fluid is traveling through the tubes, it comes in through the nephrostome, and then travels through the inside of the tubes. Then again, those tubes are made out of cells, and those cells can reabsorb water and salts and put it back into the blood through those capillaries that are branched all around the tubes. Anything that remains in the tubule, like more water and nitrogenous waste, it continues throughout the tube until it gets excreted out of the body through a Nephritio Pore again. That's the same structure, similar structure than what we saw with the previous one. Okay, so again, fluid, the body fluid comes in through the nephrostome, right, into the metanephridial system, in through the tube. As it travels along the tube, salts comes out of the tube. So salt got filtered in, salt gets reabsorbed back into the capillaries. Okay, water will follow. but not all of the water. Some of the water then remains in the tube until it gets excreted out through the Nephritio Pore, bringing along with it the nitrogenous wastes and things. Do some salts and things get excreted? Yeah. Yeah, but we're going to see when we study the vertebrates that we can control how much water, how much salt and water gets reabsorbed, and we can control if we need to release more or less and excrete more or less from our body. Okay, so for now, I just want you to see kind of a simple filtration system in a simple organism. Okay. Arthropods, and especially in this view, the insects, right, they've got a really different system here. They don't really use filtration, right? So they've got that hemolymph situation going on, right? An open circulatory system. So here they have a series of tubules called Malpighian tubules. Okay, and you can see that those Malpighian tubules actually branch off from the digestive system. Okay, but so the cells that are lining these Malpighian tubules, they will actively transport potassium ions and other salts are transported into the tubule. Now, it also actively transports a substance called uric acid. Uric acid is a type of nitrogenous waste. And we're going to talk a little bit later about that. Okay, so it's a type of nitrogenous waste. And so that actively gets transported into the tubule. Okay, so active transport means it's using energy to put it in there. Then that makes the inside of the tubule more hypertonic, right? It's got more salts, it's got more solute in there. So that means water is attracted in. So you see the arrows going in, water is also coming in and enters through by osmosis. Okay, that does not require energy. Osmosis, it's diffusion of water. Okay, so now water is entering the hindgut. Okay, but the cells also lining the hindgut, that's where reabsorption can take place. So all that salt and water that comes in, the body may determine, well, we need a balance, right? And so we brought too much salt and water in. We're going to reabsorb some of it, and the rest of it gets excreted as waste along with the feces through the anus, because again, it's attached to the digestive system. Okay, and so the uric acid, the nitrogenous waste, goes with it. Alrighty, so that's a arthropod, okay, using an insect as an example. Okay, so this will take us into osmoregulation and excretion in vertebrates. So this is going to get much more complex. And these are vertebrates that live in the water, and it can be fresh water, or it can be ocean water. And so we're going to see that there's a difference in how they deal with salt and water balance because they live in a different medium. And then we'll look at excretion of vertebrates who live on land, like us. That gets really complex. Okay, so I'm going to do that with part 2 of this video. All right, thanks guys. See you in a minute.